So now we're going to invite Susan Lin to talk to us about the Gaba receptor in epilepsy. Thanks, Susan.
Cool, thank you so much. So good afternoon all. I'm Susan, a PhD candidate at the Ion Channel Discovery Group. Today, I would like to introduce a way to assess function of epilepsy-associated GABA-A receptor variants. The GABA-A receptor is a major inhibitory ion channel of the mammalian brain, and variants in the GABA receptor is one of the causes of developmental and epileptic encephalopathy, a group of rare severe epileptic disorder. This form of epilepsy is accompanied with neurodevelopmental delay and motor disorder, with many refractive to treatment, and sometimes symptoms are even exacerbated with treatment. The current consensus is for the cause of epilepsy is that there's an imbalance between excitatory and inhibitory neurotransmission. One of the multiple ways of resulting in less inhibition is receptor desensitization, which occurs when the receptor no longer responds to the agonist. Currently, it is ambiguous whether desensitization plays a role in seizure phenotype. We therefore aim to simplify and focus on what is really going on with receptor desensitization by utilizing an equilibrium model of receptor kinetic to assess the functional parameters of desensitization, and then verify our model against 3 known pathogenic variants, as you can see here. The kinetic model that we used is this one here, so simple kinetic model. To test this model, we had to inject concatenated RNA into Xenopus oocyte and then recorded the currents 3 days after incubation. The steady state is one of the parameters we look for in our desensitization model. The steady state, you can think of it as like a plateau, so this part here. Here we have the representative traces for each group. So you have wild type and then the 3 variants here. And what we see is that there is a higher steady state current for the T287I, so T287I, and then a lower one for the R323Q. Next, we wanted to have a look at the maximum current elicited by these variants. This is the parameter we use to find how much inhibitory activity the channel has. So more current denotes more inhibition. What we can see from here is that R323Q had a significantly lower maximum current, so lower maximum current compared to wild type, and also a 1.5 times larger desensitization equilibrium with a faster deactivation here. And this is the deactivation rate, so how fast it gets off the receptor. What this means is that the variant led to a less GABA inhibitory activity. We saw no change in the maximum current in the wild type for the A332V and the T287I. So that means that the inhibitory ability is not decreased by these variant. The T287I variant did show a significantly lower equilibrium constant, but did not affect the deactivation rate compared to the wild type. Hence, overall more inhibitory activity. The result of the study indicate that only the R323Q variant increased receptor desensitization. Remember at the beginning how I said that there's an imbalance between excitatory and inhibitory neurotransmission for epilepsy, and so we would expect that the variants that increase the desensitization may show a more severe clinical phenotype. However, it did not necessarily equate to that. What we plan to do in the future is to conduct an experiment on variants that change the desensitization constant without changing the GABA sensitivity to fully understand what role desensitization has in terms of DEE pathophysiology. Thank you.
Thank you very much for that. Next speaker will be Ana Rita Barreros, who will be discussing functional connectivity in treatment-resistant depression.
Hello, everyone. Can you see my poster?
Yes, that's good.
Hear me well? Perfect. Okay, So my name is Anna Rita. I work as a research coordinator at the Brain Dynamics Centre, and I'm a PhD candidate at the University of Sydney. I'm here today to tell you about treatment-resistant depression. Concerningly, this is the reality for more than 50% of the people diagnosed with this condition. Less than 30% of the individuals diagnosed with depression achieve complete remission of symptoms. But why does it happen? We don't know. We don't know why antidepressant medication work for some people and don't. Dihabenula is a small midbrain structure associated with anhedonia, or the inability to feel pleasure, which is a characteristic of treatment-resistant depression. And it is shown to be disrupted in these patients. However, the neural circuitry of dihabenula is still unclear. This study aims to map detailed neural of treatment-resistant depression, particularly related to dihabenula. 35 patients with treatment-resistant depression, 35 patients with treatment-responsive depression, or patients who responded to treatment, to antidepressant medication, and were euthymic at the time of testing, and 38 healthy controls participated in the study. They underwent an 8-minute MRI, resting state fMRI scan at a 3 Tesla MRI scanner, and functional connectivity analysis were performed using a seed-to-voxel approach. We chose the left and the right habenula as seed regions of interest and identified them manually in the anatomic image for each participant. Whole brain voxel-wise comparisons between the 3 groups were evaluated using a significance threshold of 0.001 using FDR corrections. Demographic and clinical data summarized. However, it's very small. All the 3 groups were comparable for age, gender, and education level. And as expected, the groups were different, significantly different in some clinical variables, according to the study design. As illustrated in this figure, results shown significantly differences in functional connectivity with the 3 clusters in the brain, between the habenula and the left and right precuneus cortex, the habenula and the right precentral gyrus, which are part of the default mode network. Post-hoc tests revealed significant differences between the 3 groups for each of the 3 clusters, with evidence for hyperconnectivity in the treatment-resistant depression group in relation to the other groups. This suggests that abnormal functional connectivity in the habenula circuit might be a distinguishing factor of treatment-resistant depression. Given that the habenula receives input from the limbic system to process and encode negative feedback and negative reward, and it's part of the reward system, abnormal connectivity related to negative effect processing and reward system and its interactions with the default mode network could be a key feature for this condition. We think this is an important finding, as identifying network changes early in the brain could enable clinicians to identify treatment-resistant forms of depression and start appropriate treatments earlier. Thank you very much for listening.
Great, thanks very much, Anna. So next up we have Grace Lee to talk to us with her poster titled Right Care, First Time Where You Live. Thanks, Grace.
Thank you. Hello, and thank you for giving me the opportunity to present my research today. I'll be focusing on an evaluation study protocol on Right Care First Time Where You Live, which is a national multi-site research program that will be conducted over the next 5 years. Despite Australia being one of the first countries to develop and implement a national mental health plan in 1993, our mental health system continues to be underdeveloped and overstretched. Poor youth mental health remains a national crisis, and the youth mental health system is faced with ongoing challenges, including inefficient resource allocation. This has led to attempts by the government to invest billions of dollars in mental health systems reforms, which unfortunately has achieved little impact at a population level. Systems modeling offers a valuable tool to support strategic decision-making and can improve youth mental health outcomes at a population level, as these models can take into consideration broader social determinants of health, such as education and employment. Systems modeling is part of a larger field of computer modeling that is also used to forecast weather, simulate flight paths, and most recently has supported governments to manage their coronavirus response. It uses computer science and mathematics to simulate what-if scenarios to understand the impact of strategic decision-making, such as implementing new mental health policies and programs. The Youth Mental Health and Technology team at the Brain and Mind Center has been leading the development of systems models for mental health. What is unique about our approach is the inclusion of diverse stakeholders in the model development process, which includes young people with lived or living experience of mental illness. However, we have important gaps of knowledge, such as evaluations and systems modeling through a co-design is often conducted without a rigorous methodological approach, and in most cases, evaluations are not conducted at all. To address some of these gaps, we have developed a comprehensive multi-scale evaluation framework through the synthesis of literature via scoping review. Our evaluation framework considers 4 broad categories: feasibility, value, impact, and sustainability, with evaluation criteria broken down across the project, individual, group, and system level. Our study will also explore the diverse stakeholders' experiences, including health organization staff, health professionals, and community representatives. Systems modeling in mental health has a genuine potential to improve the lives of young people, and it has been recognized as a top innovation by the World Economic Forum this year. Our evaluation study protocol is the first of its kind, and we will deploy novel research methods, including the gamification of online surveys to conduct patient journey mapping and social network analysis. What we learn through our studies can be adapted for broader international contexts, applications, and disciplines to improve youth mental health policy, planning, and outcomes. Thank you.
Thank you, Grace. Our next speaker will be Maya Eckhart, who will be discussing the interaction of cannabis constituents. Thank you, Maya.
Hello, everyone. My name is Maya Eckhart, and I'm a PhD student at the Lambert Initiative for Cannabinoid Therapeutics. And my poster is titled Interaction of Cannabis Constituents CBCA and CBC with ABC Transporters, an in vitro evaluation. Cannabichromine, or CBC, and cannabichromanic acid, CBCA, are cannabinoids that are readily available in the cannabis plant. The state of research on them is currently quite limited. However, they have shown therapeutic potential such as anticonvulsant, anti-inflammatory, anti-cancer, antidepressant, and antibacterial properties in vitro and in vivo studies. ATP binding cassette or ABC transporters such as P-glycoprotein or PGP and breast cancer resistance protein or BCRP mediate the efflux of substrates from inside the cell to the extracellular space and are expressed in important pharmacological barriers such as the gut and the blood-brain barrier. Most importantly, it is key to know how drug interacts with them, as they have an important role in limiting the bioavailability of various drugs and are also the source of clinically significant drug-drug interactions. And so the aim of this project was to investigate the substrate and inhibitory activity of CBC and CBCA at the ABC transporters PGP and BCRP. To do this, I performed an in vitro assessment using transwell assays cultured with MTCK cells expressing human PGP or BCRP. The transwell assays mimicked an epithelial cell monolayer. And as you can see in the diagram, efflux transporters move substrates in the bisolateral to apical direction, and this movement would be inhibited by inhibitors. And so we tested compounds by adding them to one of the chambers and taking samples from the opposing chamber in order to measure the movement of compounds as a function of time in the apical to basolateral and basolateral to apical direction, and thus create measurements of compound permeability and transport ratios. Concentration of compounds were quantified using LC-MS. The results were as follows. CBCA was found to be a PGP, but not a BCRP substrate, which is displayed in the center diagram. CBC was not a substrate of either transporter. Neither CBCA nor CBC inhibited PGP or BCRP transporter function. And to discuss the results, given that CBC was not found to be a substrate or inhibitor of these transporters is favorable to its potential therapeutic development. And also the finding that CBCA is a PGP substrate in vitro could indicate a poor absorption in vivo. Further studies in vivo could translate these findings. And in conclusion, these results provide important basic pharmacological data to assist on the therapeutic development of the cannabinoids CBC and CBCA. Thank you.
Great, thanks, Maya. So our next speaker is Rianne Cicluna, and she will talk to us about the potential utility of cannabidiol for treating opioid withdrawal symptoms. So over to you, Rianne.
Thanks, Anand. Can you see my screen?
Yep, all good.
Okay, hello. Thanks for the opportunity to present today. My research is focused on the use of CBD or cannabidiol for opioid use disorder. So 250 people die every single day from an opioid overdose in the US alone, and they're on the rise in Australia too, with opioid overdoses killing more people than road accidents. Now, a key player in the development and maintenance of opioid use disorder is the development of physical dependence to opioids, characterized by withdrawal and tolerance. So withdrawal is a syndrome that emerges upon cessation of chronic opioid use, and it drives drug seeking, since the act of removing this aversive state through relapse is negatively reinforcing. The other side of physical dependence is the development of tolerance to the analgesic effects of opioids. So this is the notion whereby one must increase the dose of the opioid over time to achieve enough pain relief. Meanwhile, there's been an emerging interest in the use of medicinal cannabis products for opioid use disorder. However, most of the evidence is correlational. So in this body of research, I've examined the use of CBD or cannabidiol for opioid withdrawal and opioid tolerance. So to do this, I co-administered CBD and oxycodone for 9 days. Mice then underwent a 24-hour withdrawal period, and I tested for withdrawal symptoms. I looked at jumping behavior, which is a negative affective symptom in mice. I also looked at gastrointestinal upset. Now, to examine the effect of CBD on opioid tolerance, I co-administered CBD and oxycodone over 5 days and assessed the analgesic efficacy of oxycodone every second day. To do this, I tested how long it would take for a mouse to withdraw their paw from a heat stimulus, much like if we were to see how long it would take for a person to remove their foot off a hot pavement. So having a look at the data here, in oxycodone mice undergoing opioid withdrawal in the orange bars here, we can see that CBD dose-dependently reduced jumping behavior, that negative affective aspect of opioid withdrawal. However, we didn't observe a robust gastrointestinal disturbance in mice undergoing opioid withdrawal, and we didn't see an effect of CBD. Now, chronic co-administration of CBD actually accelerated the development of tolerance to the analgesic effects of oxycodone. So having a look at the orange bar here, this is mice undergoing opioid withdrawal that didn't receive CBD. You can see that on day 3 and day 5, they're actually withdrawing their foot more and more quickly over time. And so this is the development of opioid tolerance to that heat stimulus. Now you can see that CBD in oxycodone mice undergoing withdrawal that received CBD, you can see that it actually accelerated the development of tolerance to the analgesic effects of oxycodone. And we saw the same effect here on the cold stimulus. So this was really surprising. So CBD appeared to alleviate the negative affective symptoms that emerged during opioid withdrawal, but actually accelerate the development of tolerance to the analgesic effects of oxycodone. And so I'm now following this up with some studies to uncover the neural mechanisms behind CBD's effect on opioid tolerance. Thanks.